Thank you. I'm presenting our work where we develop tools to control reproduction in aquaculture species. So I'll start by showing why aquaculture is important. In the last 3 decades, we simply cannot harvest more from our seas and oceans. And so during this time, aquaculture has contributed to fill, the gap between supply and demand. And that trend is expected to continue because human population is obviously increasing. And so in this case, aquaculture has a role in securing seafood supply. A number of commercially exploited species of fish have become endangered, and some are even critically endangered. I'm showing here 3 of those in the top 10. And topmost of those species are the bluefin tunas. Third is the Goliath grouper, and a special type of species, the orange roughy, because they have a very long lifespan. These species are late maturing. They have a long generation time, so obviously their wild stocks are quite invulnerable to being depleted. So in this case, breeding them or breeding them in captivity is a potential way to prevent them from becoming totally extinct. So in our group, in the aquaculture group, we have worked on the southern bluefin tuna, the yellowtail kingfish, and the giant grouper. And the giant grouper is actually the focus of my talk in the slides that will follow. We focus on the reproductive axis because that's basically what drives the reproductive function in this species as in other vertebrates. So the axis is mainly comprised of the brain, the pituitary gland, and the gonad. And I have color-coded those in the photos of the fish on the left. In the brain, there is the neuropeptide we call the gonadotropin-releasing hormone, or GnRH, and that neuropeptide stimulates the pituitary gland to produce 2 hormones, follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH. And together, these pituitary hormones stimulate the gonad to develop, and ultimately the female fish will produce eggs and the male fish will produce sperm. So we take advantage of the capacity of these key hormones like GnRH, FSH, and LH in our work to manipulate reproduction in the species that we work on. Like I said, I'll be focusing on our work which we conducted on the giant grouper, or as we call it in Queensland, Queensland grouper. It's a very large species of grouper that can grow as much as 400 kilograms. Before we started our project, what we knew was that giant groupers are protogynous hermaphrodites. That means they mature first as functional females, and then they change into functional males. They are classified as vulnerable by the IUCN here in Queensland. They are a no-take species. They are heavily exploited, particularly in Asia, in the live fish food market. So their numbers in the wild have significantly declined. So obviously there is a need to breed them. Perhaps the biggest bottleneck in breeding giant grouper is basically their size, particularly at maturity. The females mature when they are about 20 kilos already, and then when they change into functional males, they're already about 40 to 60 kilograms. So I'm showing here an example. This is the facilities for our partners in Vietnam. And as you can see, one fish had to be handled by at least 2 to 3 people. So it's quite a challenge having to breed them in controlled captive conditions. So in our work, we had to deal with giant questions like, can we have smaller females, like less than 20 kilograms? And can we also obtain smaller males, like we can get them before their weigh like 40 kilograms? We were fortunate to have been funded by the Australian Centre for International Agricultural Research, which enabled our research partners in the Philippines and Vietnam to have whole or various range of or various sizes of giant groupers, especially those that were still immature. Here in Australia, we had access to giant grouper which were less than 1 kilogram in size. In addition to giant grouper, in our partner collaborators in Philippines and Vietnam, they also have stocks of smaller species of groupers like tiger grouper, which were quite handy for our as models for giant grouper for our in vivo experiments. So in generating the tools that we needed for the work to achieve or to answer our questions, we used a range of molecular techniques. I'm showing here the pipeline in which we were able to produce recombinant hormones like those that I've mentioned in my earlier slide. In addition to some molecular or advanced molecular approaches, we also use conventional methods like histology because they give us powerful data as well. So from the laboratory, we take hormones in the field to test them for biological activity, and that's under in vivo conditions. So perhaps the most important tool that we generated are the recombinant hormones, and we have two versions of those. A version which we can administer by injection, which is the traditional way of hormonal treatment in fish. And we have recombinant hormones that can be administered via the oral route. And basically the recombinant hormones are incorporated in the feed and then given to the fish. And this is very advantageous for dealing with a large size species of fish like the giant grouper. We have generated various assays, ELISAs, non-invasive sampling methods, and I would like to highlight here the ELISA which we developed for vitellogenin. Vitellogenin is the protein that gets incorporated into the eggs, so it is a marker for mature females, and normally it is measured from plasma, which means we have to collect blood, and that's rather stressful for the fish. So what we did was to develop an ELISA that would measure vitellogenin from skin mucus. So basically we have the fish and then scrape off a bit of mucus from the skin and run that on that ELISA which we developed. And indeed we were able to validate that the ELISA works because we can discriminate mature females that showed high levels of vitellogenin in the mucus against a male where the vitellogenin basically was undetectable. We also generated a lot of other tools like genetic markers, which is important for sex-changing species, transcriptomic data, antibodies, and others. These are important not only for developing breeding techniques, but we use those to further understand biology or specifically the reproductive biology of the species that we're working on, because at the beginning we don't really know everything about them. So did we manage to answer our giant questions? Absolutely. I'm showing here the histology of the gonads of fish which we treated with recombinant giant grouper FSH. So this studies was actually done in tiger grouper. So there were two groups of fish, and these were immature at the beginning of the experiment, and the treated group were fed with recombinant FSH, and the sequence of the FSH was based on the giant grouper sequence. So the fish were fed once a week for every week for a period of 38 weeks. So just on the 8th week, there was a significant development in the ovaries of the treated fish, much more advanced compared to the control. And at this stage, this is already similar to the control after 20 weeks when the experiment started. And at this point, the treated fish were already transitioning to males, and that developed further on the 38th week when we had to terminate the experiment. So based on these results, we calculated that for giant grouper, we can obtain functional females that are about 10 kilograms and functional males, which are about 20 kilos. And that's a big advantage for the industry. So in the course of our work, we also discovered that giant grouper, in fact, is exhibiting diandry. So what does that mean? There are individuals that mature as usual, like protogynous hermaphrodites that mature first as functional females and then change into functional males. But there are a few in a cohort that develop directly as males, so they don't go through the functional female phase. And in the stocks in the Philippines and Vietnam, in our collaborating institutes, about a third in the cohort are functional males. So this is quite a significant discovery for us, because combined with our recombinant FSH treatment, we can predict that we would be able to obtain even smaller functional males, maybe even smaller than 20 kilos, let's say 10 to 15 kilograms. So what are the other applications of the tools that we have developed? Breeding other species, but not necessarily just fish. We had a short project before on the green and golden bell frog, which is an endangered species in New South Wales. The University of Newcastle has been trying to breed them in captivity, but their results have been inconsistent. So actually their partners in Canada from University of Ottawa approached us to develop some or generate some tools for them, and we have, and hopefully they would be able to use those in their future work. We are starting a project on the Macquarie perch, which is a threatened freshwater species in New South Wales. They are now have become rare in their native habitats, so there is an effort to breed them in captivity to be able to generate populations that can be put back where they used to live. Or perhaps the most recent biggest success we had was in the white teeth fish. This isn't a fish, this is a sea cucumber, which has become very rare as well in the wild. We generated tools and we used similar approaches, and we have been very successful in making them breed in captive conditions. So in summary, basically our tools are quite significant for aquaculture purposes, but in addition, they do make impacts in other areas as well, like in efforts towards conservation of endangered species. So, like I said earlier, with a group work on the giant grouper, we were funded by ACR. We are led by Abigail in the work on fish reproduction. We had students in the project. Some have finished already. We had collaborators in other countries. A lot of people, staff in the lab and GeneCology have helped us somehow in our work. So we acknowledge them and thank them very much. Thank you.
Thank you very much, Josephine. Are there any questions for Joe? There's a question from Susan. Does the acceleration of the females to male also initiate other changes such as tumor growth?
We haven't looked into that. Yeah, perhaps that's something that we can look at as a side study in the future, but we were just focusing on reproductive function, so we don't really know.
I think because it's stimulating like semi-natural hormones rather than excessive growth or something, we hope it wouldn't cause, but that is a good point, an interesting one to have a look at. All right, thank you, Joe, that's great. And we now move to Fraser, please. Yes, we can see that. Great, thanks, Fraser.
Good. Okay, well, thanks very much, Abigail. What I'd like to do today is talk about a project that's really been on and off over the last couple of years, and this is to do with identifying a scar tissue remedy from stingless bee cerumen. And the reason that we're interested in this is that we think that there may be molecules, we hypothesize that there may be molecules within stingless bee cerumen.
Fraser, your slide disappeared, sorry. Okay, now we can see. If you want to move to presentation format, it's in. If not, that's okay too. That's fine.
How do I do that?
In the PowerPoint, but that's okay. I mean, it doesn't matter. Yeah, there. Yes, thank you.
Perfect. Sorry about that. All right. Yeah, as I was saying, we're interested in the potential of the stingless bee cerumen to be able to treat chronic wounds. So chronic or non-healing wounds are important because they affect around about 430,000 Australians annually. And this incurs a cost to the healthcare budget of Australia to the tune of about $3.5 billion. Hypertrophic scarring is an aberrant form of healing of chronic wounds, and it's a fibroproliferative disorder that's associated with excessive deposition of extracellular matrix into the wound region. And there are treatments for this. So a first-line treatment, in fact, is corticosteroids, but this is very much limited because it can cause atrophy of the tissue and it can also break capillaries. So we think that new strategies are needed. So we've focused on Tetragonula carbonaria, which is a species of Australian stingless bee, and you can see in the top left image, this is a photo that I took in the front yard of our house. It's a stingless bee that is collecting nectar, and it uses this as an energy source. The image directly below that is from a paper published by Helen Wallace, and it shows the stingless bee collecting resin from trees, and what happens is that the bee adds its own salivary secretions to the resin to produce a product called cerumen, and the bee then deposits that cerumen into its hive. And it's thought that it does this because of the protective effects that the cerumen has against pathogens. So there are known antibacterial properties, for example, to cerumen. The image on the right shows a hive, and this image is courtesy of Stephen Truman, and it shows the brood in the middle. Down the bottom are some pots containing pollen, which is a protein source for the bees. Up the top you can see some honey. So these bees also produce honey, albeit to a much lesser extent than the European honeybee counterpart. And what we're interested in is this really dark brown substance on the right-hand side, which is the cerumen. So what we've done is taken this stingless bee cerumen, and we've run an initial purification using solvent extraction to generate a crude solvent extract. And this work was carried out by Dr. Karina Hamilton as part of her PhD. So what Karina did was she injected the solvent extract onto an HPLC column, and the purpose of this is to separate the molecules according to their polarity. So we can get individual compounds, and you can see the trace down the bottom here with the HPLC trace with the individual peaks. So these represent one or maybe a small handful of compounds that have been separated. So we undertook a program of what is referred to as bioactivity guided fractionation, and I note that some others have already talked about this type of approach in the earlier session. So using the bioactivity guided fractionation approach, we were able to ultimately isolate a single bioactive molecule, which we dubbed compound 35, and we had carried out studies to elucidate the chemical structure of this molecule, which is currently subject to patent application. So the first experiment was really to see whether compound 35 might be able to attenuate fibroblast proliferation. And this is important because of the role that fibroblasts have in the wound healing process. So we cultured the fibroblasts at a level of 20% confluence and then incubated the cells in the absence or in the presence of increasing concentrations of compound 35 or the vehicle, which in this case was DMSO. And the results are shown over on the right-hand side. We can see here that compound 35 caused a dose-dependent inhibition of fibroblast proliferation. So following on from that, we wanted to see whether compound 35 might also modulate cellular migration into the wound region. And so we established a scratch assay whereby we grew fibroblasts to subconfluence level, and then we created a wound by simply dragging a pipette tip across the surface of the monolayer. And I hope you can see this. This generated a cleared region in the middle where there are no cells. So what I'm going to do is I'm going to show you some time-lapse microscopy where we will visualize the regrowth of the fibroblasts into that wound region over a 72-hour period. So these images were captured at 2-hourly intervals, and you can see that there is not only proliferation but migration of the cells into that wound region. So we repeated this experiment again in the absence or presence of compound 35 to see whether it might have an effect, and the results shown there on the right-hand side. Compound 35 caused a dose-dependent inhibition of fibroblast cell migration. And interestingly, we saw similar effects on keratinocytes as well. So that's all very well. That is in line with our hypothesis that compound 35 might be useful in the treatment of scar tissue formation. We know that during scar tissue formation, fibroblasts need to differentiate into myofibroblasts, which then generate collagen that is deposited into the wound region. So we wanted to test, therefore, the ability of compound 35 to interfere with that process. And we examined this by measuring the expression levels of smooth muscle alpha-actin, which is a biomarker of myofibroblasts. So this differentiation occurs under the influence of the cytokine transforming growth factor beta-1 or TGF-beta-1, and we're looking at expression levels of smooth muscle alpha-actin. And this work was carried out by Daniel Tchaikovsky as part of his honours program. This is a confocal microscopy image of fibroblasts that were incubated in the absence of TGF-beta1 and labelled with an antibody directed against smooth muscle alpha-actin, which we see in green. The blue bits that you see here, these are the nuclei stained with DAPI, which is a nuclear marker. So what we conclude from this is that the fibroblasts normally have a low expression level of smooth muscle alpha-actin, and that this is primarily distributed in the perinuclear region. And if we look at the fibroblasts then after the incubation with TGF-beta1, we see a vastly different picture. So we get these beautiful green striations coming through, which is the smooth muscle alpha-actin on the cytoskeletal structure. So this is consistent with the ability of TGF-beta1 to transform the fibroblasts into the myofibroblasts, the collagen-producing myofibroblasts. So the next thing then was to see whether compound 35 could prevent that. We incubated the cells then with TGF-beta1 and compound 35. So just to orientate you, this is what I showed you before, without TGF-beta1, with TGF-beta1, and then on the right-hand side, this is the interesting data, with TGF-beta1 and co-treated with compound 35. So we're able to inhibit the formation of smooth muscle alpha-actin, so consistent with our ability to block the transformation of fibroblasts to myofibroblasts. So if our thinking is correct, compound 35 should also be able to prevent collagen production by the fibroblasts. And this was examined. We incubated the cells with TGF-beta1 in the absence or presence of compound 35, and we saw a significant reduction in soluble collagen production by the cells. So that's where we're at the moment. The next phase of the study is going to be to file a patent application. We think that this molecule may well be of interest in terms of a possible candidate for treatment of hypertrophic scars, and also to run a preclinical trial in which we administer compound 35 to an animal model of hypertrophic scar formation. So this is very much a collaborative project, and there's been enormous contributions from Steve Ogborn, Peter Brooks and Trong Tran, and I'd like to acknowledge the great work done by Karina, Daniel and Nick Kong as well, and our collaborators from the US, UK and Brisbane.
Thank you, Fraser. That's been fascinating and lovely to see. It looks amazing. So with, I guess, with patents and then taking it into any clinical trials, what's the path? I mean, patent, yeah, I know, but you need to have an industry partner, a medical partner that would be interested in then sponsoring
yes.
the trials, or?
Ideally, Abigail. In fact, probably more than ideally, that would be an essential part of the progression of this project. So we're looking to run, hopefully, the animal study in the next 12 months, and to use that preclinical trial data to then to be able to go after some larger funding that would be required to take this idea further.
You have, thank you. You have a question here from Lockie about the if it has any other effects on the cells or only mediation of TGF-beta1.
Yeah, thanks, Lockie. We did run an MTT cytotoxicity assay. There is a window, I'm going to say a window of opportunity with this compound that if we go up to very high concentrations, we start to see some cellular loss, but that wasn't the case at the concentrations that I used and showed in this presentation. So for example, in the confocal images, we still saw viable cells, and that was evidenced by the presence of the blue staining for the nuclei.
Fraser, you might want to read, there's another couple of questions in the chats for you from Carla and from Bonnie.
Okay, I'll just get to the chat.
You can see them. If not, I'll read them for you.
I'm just trying to find the chat.
That's okay. Carla asks, how did you first determine compound 35 as a candidate compound with potential to have these effects as, you know, compared with other compounds?
Yeah, in this process of bioactivity-guided fractionation, we started off with something like 200 compounds, and we screened that to initially identify that there was some bioactivity of interest, and then through the process of HPLC fractionation, we narrowed that down to a much smaller number of compounds. So, you know, setting up several fractions, and we tested each of those fractions, and then found that there was a small cohort of compounds that still had the bioactivity. And we just kept repeating that process over and over until we eventually came down to a single molecule. And that's the one that we're running with.
Thank you. And last question from Bonnie. Moving to a mouse model, do you have an idea about the toxicity of the compound or dosages to see an effect?
Yeah, right. So I think we'll learn a lot from the animal model. We propose a mouse model of hypertrophic scar formation. So this involves removal of a small segment of skin from the back of the animal and surgical implantation of some human skin to replace that. And that's been shown, not by us, but by others, to be a reasonable animal model. So what we propose to do is to apply the compound 35 superficially to that wound and see whether it might be able to limit hypertrophic scar tissue formation. So to answer your question, Bonnie, we ought to be able to gauge whether there are any cytotoxic effects in the animal model by running several doses of the compound.
Thank you, Fraser. I think Tim Wes would have particularly enjoyed your talk, being Mr. Collagen.
Yeah.
And I think this is another example, and I mean all of your talks are, but another example how I think the powers to be or not to be, the existing powers really want to separate medical research from, say, GeneCology, and for like John Bartlett, really didn't want to have anyone in medical research associated with the center because they said, no, that's separate. And I think this is just a beautiful example why that's not separate at all. And look how the world of stingless bees translated into, you know, such a beautiful project. So
Yeah.
It's fantastic. Thank you, Fraser.
Thanks, Abigail.
If you can stop sharing your